Welcome to the Wednesday afternoon lecture. Today we have the honor of having with us Dan Littman, who's going to talk about a very interesting interaction between the microbiome and the immune system. He is an investigator at the Howard Hughes Medical Institute and coordinator of the Molecular Pathogenesis Program and Kimmel Professor of Molecular Immunology at the Skirball Institute at NYU. Dan got his undergraduate degree at Princeton and got an MD PhD in molecular biology at WashU. Following that, did a postdoctoral time with Richard Axel at Columbia, and then set up his own laboratory at UCSF, where for the next 10 years he was assistant professor, associate professor, and then full professor. But since 1995, he's been at the NYU School of Medicine, where he is both investigator of HHMI, but also, as I mentioned, Kimmel Professor. He's been recognized by a number of honors, notably election to the National Academy of Sciences in 2003, and the Ross Prize, the Vilsack Prize, and the Coley Prize. And his investigations that he's going to tell you about understand ways in which T lymphocytes acquire functional properties during development in the thymus and how this is influenced by signals initiated by distinct commensal microbes in the intestine. And so here we are at a time where I think one would be tempted to say, and would say correctly, that there is a real revolution going on in our understanding of the microbiome and our abilities to be able to assess what's happening, particularly in the GI tract. But there also seems to be a period of rapid advances in immunology, and to put the two together is another great example of how sometimes the most exciting science happens at those interfaces between disciplines. And there could be nobody better to represent that this afternoon in this space than Dan Littman. So please join me in welcoming Dr. Dan Littman.
Thank you so much, Francis. And it's really a great honor to be invited here. Thank you so much for having me. I have a lot of very warm feelings for the NIH for many reasons. I was here in 1976, just as I was starting my PhD work as part of my MD PhD, because my advisors were finishing their postdoctoral fellowships here, and they were in transition to St. Louis, and I helped them with the move after spending the bicentennial summer in Washington, which was really a very special time. So as Francis mentioned, I will tell you about our work on trying to deconvolute how microbiota interacts with the immune system, and in particular with T lymphocytes. And the title that I have here, I'm not exactly, certainly we think the microbiota act as in instructing T lymphocytes as to their fate, but also in deciding whether, when they are on their way to one fate or another, which branch they're going to take. And there's a lot of plasticity in the immune system, and I'll tell you a little bit about the various options. Now, we've heard, of course, a great deal about the microbiota during the past 5 to 10 years. And we now know that the commensals within us contribute to insulin resistance or sensitivity. They have a very important role in obesity. There's beautiful work from Jeff Gordon's group, who's been a pioneer in this area, in showing that there are microbiota that in twins who are discordant in terms of their weight, that can basically predispose mice to the same phenotype as the human host. There are also examples, I'm not sure if this is working okay. There are also examples of the microbiota influencing the central nervous system and behavior, and I'll tell you a little bit about an example of this towards the end of my talk. And then the microbiota have even been implicated in the diurnal clock, in circadian rhythm. And more recently, a great deal of exciting work, including work coming from here, from the Trincheri laboratory, has shown that commensal microbes can influence responses to cancer therapy, both chemotherapy and immunotherapy. And we still have a very poor understanding of what's going on there, but we think that some of the work that we and others are doing to try to understand responses of the immune system may contribute to eventually gaining an understanding there of how these microbes might be manipulated in treatment of cancer. Now, our entry into this field comes via a molecule called ROR gamma T. It's a transcription factor that is a nuclear receptor similar to glucocorticoid or estrogen receptors. It has ligands in the cholesterol family, in the cholesterol biosynthetic pathway, although the precise ligand, I think it's fair to say, has not been identified. We think there are multiple ligands. And this is a molecule that we discovered years ago as being involved in the thymus for survival of immature thymocytes that give rise to the CD4 and CD8 cells in the periphery. At the time that we started working on this, we also found that it has a role in lymphoid organ development, in the development of Peyer's patches and lymph nodes. But the real excitement in this field came subsequently when it was found that ROR gamma T has a role also in certain classes of T cells and of innate lymphoid cells, the type 3 innate lymphoid cells. And I'll tell you a bit about each of these different cells, not so much about gamma delta T cells. And what I will mostly focus on will be the Th17 cells because it was really the discovery of these cells by several groups, most prominently Dan Kua at DNEX, that really set us on the way, as I will tell you about briefly here. So when we first started working on ROR gamma T and realized its role in induction of lymphoid organs, we found that it's expressed in a cell that was first described by Raina Mebius and Irv Weissman that eventually was named the lymphoid tissue inducer cell. And this is found in the fetus for development of lymph nodes and Peyer's patches, secondary lymphoid organs. But it's also found postnatally in tertiary lymphoid organs, such as this structure that occurs in the intestine just underneath the epithelial layer that's shown over here. And this is called a cryptopatch. And what this cryptopatch consists of are these ROR gamma T positive innate lymphoid cells. And it's surrounded by dendritic cells. And it senses the state of the microbiota across this single cell epithelium. And upon signaling from the microbiota, recruits B lymphocytes to become an isolated lymphoid follicle. And Gerard de Berle, who was a postdoc in the lab, made a knock-in of GFP into the ROR gamma T locus. In homozygous knock-in mice in which the product is null, there are no such structures being formed. But in a heterozygous mouse, we can follow the expression of the GFP in cells that express a normal copy of ROR gamma T. And Ivo Ivanov, a new postdoc at the time in the lab, began to look at these structures in the intestine and made a discovery we had missed earlier, and that is that there are not just these bright GFP-positive cells that are innate lymphoid cells that don't have any other kinds of hematopoietic markers for lineages, but there are also these intermediate GFP population of cells here that are T cells that express the alpha-beta T cell receptor. And around the same time, I received a call from Dan Kua, who told me that they were looking at these mysterious new cells that were responsive to interleukin 23. They would turn on production of IL-17 in response to IL-23, and they found that one of the genes that was upregulated was ROR gamma T. So in collaboration with Brent McKenzie in his lab, in Dan Kua's lab, we began looking at these cells, and very quickly were able to show that only in these GFP intermediate cells, there was also a lot of expression of intracellular interleukin 17. In the GFP ROR gamma T negative T cells, there was no expression of IL-17. And this was really the way that we got into this field, because we were able to very quickly show then that in animals that were null for ROR gamma T, there was very little, if any, interleukin 17 made by the T cells in the intestine. And we also recognized at that time that these Th17 cells were most abundant in the intestine. So it had already been known since the mid-1980s about Th1 and Th2 cells, and I don't need to tell this audience about this. The late Bill Paul was a pioneer in this area, was really one of the heroes in the area of T-cell biology, and Bill's laboratory did a great deal on characterizing the molecular features of these cells. The Th1 cells are dependent on the transcription factor Tbet, and they make interferon gamma. They are generally required to kill intracellular pathogens, but they've also been implicated in autoimmune diseases. And the Th2 cells are dependent on the transcription factor GATA3. They make IL-4, IL-5, and IL-13. They're very important for controlling infection with parasitic worms, with helminths, but they also contribute to allergy and to asthma through the production of these cytokines. And these were the cells that had been thought to be the major types of helper cells until the mid-2000s. And then with the advent of the discovery of cells that make IL-17 that are dependent on ROR gamma T, it became clear that these cells make several other cytokines, including interleukin-17F and interleukin-22. And it was recognized that these are cells that are required to control extracellular microbes. I wrote here pathogens. It's an old slide, but not only pathogens, but also commensal microbes, including both bacteria and fungi. And the cells are very important in repair of tissues at barrier surfaces, primarily epithelial surfaces. And they've also been implicated in autoimmune inflammatory diseases, and that's why there's been an enormous amount of attention paid to these cells. And I'll tell you a bit later about the various cytokines that are required for the generation of these cells. And then a fourth type of CD4 positive T cell is the regulatory T cell. The Treg cells develop in the thymus as so-called thymic-derived or natural Tregs, but there are also peripheral induced Tregs that express FOXP3, are dependent on the transcription FOXP3. And all of these cells are really essential for restraining these other types of T cells and preventing autoimmune diseases. And as I'll show you, the induced Tregs in the intestine also express ROR gamma T. So the Th17 cells are the ones that have really been at the center of studies of autoimmunity during the last half dozen years or so. And I told you already about their importance at barrier surfaces in controlling infections with these pathogens here. But these cells have also been implicated in these different kinds of inflammatory diseases. I should say that psoriasis is probably the best validated because antibodies against interleukin-17 and interleukin-23 are tremendously effective in therapy for psoriasis. However, that targets IL-17 made not only by the Th17 cells, but also very likely by gamma delta T cells. But some of these other diseases also appear to be Th17 related. A lot of arthritides, spondyloarthropathies are dependent on Th17. Rheumatoid arthritis likely has a Th17 component. Inflammatory bowel diseases are very much dependent on interleukin-23 signaling and probably a combination of Th17 cells with some of the other cells that make interleukin-17. And probably steroid-resistant asthma as well as MS have Th17 components. And I put here as a question mark autism spectrum disorder because I'll tell you about a model in mice in which, certainly in that model, Th17 cells appear to have a role. So this is just an example then of what we were able to show as soon as we understood that Th17 cells were most abundant in the intestine, in the lamina propria. In our colony, they happen to be most abundant in the small intestine. It's not true in all colonies. It really depends on the type of microbiota present there. But what we found was that when animals were treated with antibiotics or re-derived as germ-free, there were essentially no IL-17A producing cells in the intestine in these mice. And this is work, again, of Ivo Ivanov. And then Ivo made an observation that really took us into studying the microbiota. What he found was that the same strain of mice obtained from different sources differed in the proportion of Th17 cells among CD4 cells in the intestine. So mice from Jackson Laboratory had very few such cells, 1 to 2% of CD4 cells, whereas mice from Taconic typically had 15% or so cells making interleukin-17A. And what he was able to show, with help now from our collaborator Kenya Honda in Japan, was that this was simply due to a difference in one single bacterial species, and that is due to the presence of segmented filamentous bacteria in mice from this source. And this is a gram-positive spore-forming anaerobic bacterium, very different from other taxons of bacteria that have been described, but probably if it's related to anything, it's most closely related to the Clostridia. But these bacteria, you can see, form these long filaments that cover the villi in the ileum. And they're completely absent and still absent in most mice obtained from Jackson Laboratory. And then you can see that in a transmission EM here, that these bacteria form these segments, and they embed themselves in the epithelium of the ileal, in the ileum, in terminal ileum, and cause an actin polymerization reaction. We still understand nothing about any kind of molecular mechanism involved here. But we're able to obtain fecal material from animals that were mono-associated with SFB, and gavage germ-free mice with these. And you can see the germ-free mice here have essentially no production of IL-22 or IL-17 in their CD4 cells. And within a week of gavaging with SFB, you see this massive induction of these cytokines. And what this does is to help protect the mucosal barrier from pathogenic bacteria, but at the same time, it also can make the animals more susceptible to developing spontaneous arthritis in a mouse model that we studied in collaboration with Diane Mathis and Christophe Benoit. So this is an example of the protection from an adhering effacing bacterium, Citrobacter rodentium, which adheres to the colonic epithelium. And you can see when mice are colonized with SFB, there's considerably less adhesion, and there's also less weight loss in these animals colonized with Citrobacter. On the other hand, the flip side is that in the KB/N mouse model of arthritis, in which Diane and Christophe had shown that there is no disease when the animals were re-derived to be germ-free, colonization with SFB alone within only a few days now leads to thickening of the joints and to inflammation and to arthritis. And this is work of Joyce Wu in collaboration with Ivo Ivanov in our lab. And what Joyce was able to show is that even though this disease is dependent on an antibody response and an antibody against a self-protein, that it requires Th17 cell polarization. And indeed, this is what SFB is achieving here. So this led us to consider that there may be something similar in humans with rheumatoid arthritis as to what had been observed in this animal model. And Jose Scher at the time was a rheumatology fellow at NYU. He's now on the faculty. And Jose decided to look at cohorts of patients at Bellevue Hospital who had either new onset rheumatoid arthritis, NORA patients, who had not yet been treated, or chronic arthritis patients who had undergone treatment for a number of years, and healthy individual, and also psoriatic arthritis. And we performed initially 16S ribosomal RNA gene sequencing to look at the profile of microbiota in these individuals, and then subsequently followed that with metagenomic deep sequencing of the microbiota. And what we found is that more than 70% of the new onset arthritis patients were colonized with one type of gram-negative bacterium, Prevotella copri. And this was found typically in the healthy population, and in chronic arthritis patients in only 15 to 20% of these populations. Now, we still don't know whether Prevotella copri has a causal role. Obviously, SFB appears to have a causal role in the animal model, and one of our goals is to determine if there are bacteria that have these kinds of functions in human disease. But what we were able to show in our early studies was that when we did comparison of the metagenomic sequences from patients and from healthy individuals, we found that there were certain clusters of genes that differed between them in very significant, significant fashion, suggesting that perhaps there may be virulence genes in these particular strains of Prevotella. So we're currently have been growing out the Prevotella strains from healthy and affected individuals and trying to compare them in animal models to look for causality. But the work that I was telling you about begs the question of how it is that bacteria that colonize the intestine can lead to systemic autoimmunity, to systemic diseases such as arthritis shown here. So we began to look at what are the properties of the Th17 cells that are induced by SFB. And this basically is a summary of what we have found, and that is that the bacteria that induce SFB do so in the lamina propria, that the bacteria that induce the Th17 cells do so in the lamina propria, but the polarization occurs in the draining lymph node, in the mesenteric lymph node. And these T cells that respond are specific for antigen encoded by SFB, and that is presented by MHC class II. The very same antigen can be manipulated to be expressed in a different bacterium that induces Th1 cells. And Listeria monocytogenes is such a bacterium. And what we found is that even though the T cells have the same specificity as for SFB, these become now Th1 cells that are polarized by Listeria. And as I'll show you in a moment, there are also bacteria that polarize cells towards regulatory T cells, the induced Tregs. Kenya Honda was first to show this with a number of different Clostridial species, both from mouse and from human, that when introduced into the mouse GI tract in the colon led to induction of regulatory T cells. And as I'll show you, many of these regulatory T cells are specific for the microbial antigen that elicited these. So just to show you that work, actually this is work done by Mo Shu, who's a postdoc in our lab. And Mo compared induction of T cells by segmented filamentous bacterium and Helicobacter hepaticus. Now SFB, I told you about, induces this Th17 response under what we consider to be homeostatic conditions. There's no sign of inflammation in these animals. The Th17 cells are abundant, but they do not promote colitis, and I told you that these are found typically in the ileum, in the terminal ileum, the SFB colonization. Helicobacter hepaticus, on the other hand, is in the colon primarily, and it can induce a pathogenic type of T cell response in models of colitis. So Fiona Powry has pioneered this field, and she has shown that introduction of naive T cells into animals that lack any T or B cells in rag knockout mice leads to a colitis that is dependent on a mixed Th17, Th1 response, that is cells that make both IL-17 and interferon gamma. But we wanted to see what is the T cell response under homeostatic conditions. Now, in her case, this kind of a colitis required that there be colonization with bacteria, most often Helicobacter hepaticus. Also, if one blocks IL-10 production in animals colonized with Helicobacter, there's a similar inflammatory colitis. So what Mo decided to do then was to isolate T cells from the intestine of mice colonized with Helicobacter and identify the T cell receptors on these cells that were elicited by Helicobacter, and made T cell receptor transgenic mice, as well as generated MHC tetramers to be able to track these particular T cells, which turned out to be specific for Helicobacter antigen. So he used now a combination of T cells specific for SFB and T cells specific for Helicobacter in animals that were simultaneously colonized with both of these bacteria. So he's able, using isotype markers, to distinguish between the T cells specific for either of these bacterial antigens. And I show you a comparison between these here, because now when he recovers the T cells from the lamina propria in these mice, you see that the 7B8 cells that are specific for SFB all become ROR gamma T positive cells. And these are Th17 cells that are non-pathogenic, as I told you. On the other hand, the Helicobacter-specific T cells, you see by and large become FOXP3 positive cells, shown over here, which also express ROR gamma T. So this is the hallmark of these types of T cells that differentiate in the intestine to become regulatory T cells, and that have been shown to have unique properties different from other kinds of regulatory T cells. The T cells that do not express FOXP3 and ROR gamma T express the chemokine receptor CXCR5 and the transcription factor BCL6, and these are follicular helper cells that are involved now in the production of antibodies. So what happens when these are introduced? These cells are introduced into IL-10 knockout mice. So I told you that in IL-10 knockout mice, colonization with Helicobacter leads to colitis. And what we see now is that these cells now differentiate into ROR gamma T positive cells for the most part. And unlike the cells specific for SFB, these cells are pathogenic. And I'll explain to you what that means in a moment. Well, first of all, let me show you what happens when we look at IL-10 knockout mice versus IL-10 sufficient mice colonized with the two bacteria and now repopulated with these two different types of T cells. You see that the Helicobacter-specific T cells express not only IL-17 on the x-axis, but also interferon gamma on the y-axis. So this is one of the hallmarks of the pathogenic T cells. On the other hand, the SFB-specific T cells, even in the IL-10 knockout, only express interleukin-17A and no interferon gamma. And when we've done RNA sequencing to compare these populations, also single-cell RNA-seq, we see that there's really hardly any difference in the presence or absence of IL-10 of these cells, whereas the Helicobacter-specific cells change dramatically in their properties after exposure to the IL-10 deficient background. So this takes me to the topic of pathogenic Th17 cells in these mouse models, and whether there may be such cells in human. And in fact, we think that there may in fact be such cells in human, and they've been called Th1 star cells by Federica Salusto, who first described them. These cells in the mouse are dependent on the interleukin-23 receptor. So I told you that early on, Dan Kua showed that IL-23 signaling leads to upregulation of interleukin-17 in vitro, and it's required for pathogenesis in various mouse models of autoimmunity. In particular, there was an experiment done by Gita Stockinger's group in London showing that using fate mapping, that many of the cells that initially had expressed interleukin-17A went on later to express interferon gamma, and that process was dependent on interleukin-23 receptor. And only in the presence of interleukin-23 receptor could these cells be pathogenic in a model, in autoimmunity model of experimental autoimmune encephalomyelitis. And we think something very similar may be occurring in human, because Federica had described cells that express the chemokine receptors CCR6, which is a Th17 related chemokine receptor, as well as CXCR3, which is a Th1 chemokine receptor downstream of the transcription factor T-bet. And these double expressing cells also produce interferon gamma, and these are cells she called Th1 star. And Jean-Laurent Casanova at Rockefeller found several patient families that had immunodeficiencies, that had deficiency in ROR gamma T. They're basically null for ROR gamma T, and these cells were lacking, but these individuals still had classical Th1 cells making interferon gamma in response to viral infections. Both Federica and Jean-Laurent then also found that these are cells that are particularly responsive to mycobacteria. So if these individuals from these families lacking ROR gamma T were inoculated with BCG, they developed disseminated BCG infection, and a couple of them did not survive. And that's because it seems that these Th1 star cells are required for controlling this type of bacterium, the mycobacteria. So what is special then about these inflammatory pathogenic Th17 cells? Well, Lin Wu has taken a different approach to look at these, and that is to use rag knockout mice in which he reconstitutes bone marrow from IL-23 receptor sufficient and IL-23 receptor deficient mice, and then subjects these animals to a couple of autoimmunity models as well as colonization with SFB. And the autoimmunity models are EAE, to look at central nervous system infiltrates, a colitis model with antibody against IL-10 or IL-10 receptors I already told you about, and the SFB colonization I told you about. And then he did single-cell RNA sequencing in collaboration with Raul Satija, who's at NYU and the New York Genome Center. And looking initially at CD4-positive T cells from the central nervous system in mice in which there was induction of EAE, he was able to cluster cells into different categories as shown here. And importantly, this category over here had a lot of cells that had FOXP3 and interleukin-10, whereas this category over here had cells that made IL-17A, interferon gamma, and also GM-CSF or CSF2. And these are properties of what have been described as these inflammatory pathogenic Th17 cells. And when he looked then at distribution of IL-23 receptor sufficient and deficient cells, what he saw was that the deficient cells were underrepresented in this pathogenic population here, but the IL-23 independent cells were enriched here in what we might call a regulatory type population that we don't think contributes to the disease. So what happens then upon colonization with SFB? Well, if we now look in the intestine at CD4 cells, what we see is that there are regulatory T cells that express FOXP3, which are cells over here, which fall into the category of those that also make ROR gamma T, and those that make GATA3, which we think are thymic-derived cells. And then there are what might be considered to be more classical, conventional Th1 type cells that make interferon gamma over here, but there are no cells that make both interferon gamma and IL-17A. You have instead these, what we'd consider to be these homeostatic Th17 cells down in this category over here. So we're obviously very interested in trying to understand what regulates these cells going one direction or another. Vijay Kuchru has done some really beautiful work in this area in which he has identified several candidates that may be involved in this process. We're still trying to understand what the relationship is between these induced regulatory T cells and the Th17 cells. And we'd like to know, we know that IL-23 is required for these pathogenic cells to differentiate. We think that they differentiate from what might be a homeostatic Th17 cells. We know that IL-10 restrains this differentiation. And we don't really know exactly yet where these Treg cells come from, although we think that they likely polarize from naive CD4-positive T cells. And we also know that these cells are absolutely required to restrain this process of expansion of pathogenic Th17 cells. We have data in which if we completely eliminate a transcription factor in these cells, we're able to see the expansion of these pathogenic Th17 cells and see spontaneous colitis. So coming back then to what might be occurring upon colonization with bacteria, what I told you then is that with SFB, there is this induction we see of the Th17 cells, and we see the induction occurring temporally first in the draining mesenteric lymph node within the first 3 days. And then a few days later, these cells are found in the lamina propria, where they are making interleukin-17A and F and IL-22. But we also see these cells distributing quite broadly throughout the other parts of the intestine to distal lymph nodes and also to spleen. So we think that may in part explain why T cells that are elicited here in the gut mucosa by microbiota may contribute in some ways to autoimmunity. And that has really been our goal, to try to understand what it is that can trigger autoimmunity in animals that are colonized with certain bacteria. So to do this, we started taking a closer look at this induction of ROR gamma T and IL-17A, doing timed colonization with SFB. And I told you already that SFB is mostly in the terminal ileum. This is a log scale of colonization. and you can see in mice from Jackson Laboratory, we don't see any SFB at all. And we also see a change in gene expression patterns that seem to be confined to the ileal epithelium. And the genes that are most highly upregulated in the epithelium are the serum amyloid A proteins 1 and 2. And I'll tell you more about SAA in the next part of this talk. And you see this occurs in the ileum, but not in other parts of the intestine. And then Teruyuki Sano, who did this work, made a very interesting finding that's shown over here. It was a bit surprising. And that was that when he looked across the intestine, duodenum, ileum, and colon shown here, he saw similar proportions of ROR gamma T positive CD4 T cells, what we consider BTH17 cells, across the gut. But it was only in the ileum that he saw that most of the cells also expressed interleukin-17A. This is using a reporter mouse, so we don't have to stimulate the cells ex vivo. We can just take them directly out of the lamina propria and look at GFP expression. So we wanted to know what is it about the ileum that's special that allows induction of the effector functions here. And he performed a series of genetic experiments and also antibody blocking experiments. One experiment is shown over here in which we focused on the serum amyloid A proteins. We're able to use these T-cell receptor transgenic T cells that are specific for SFB and introduce them into mice that either were not colonized with SFB or were colonized with SFB. So the cells were labeled with the intracellular dye CFSE so we could look at cell proliferation. You can see that if we now look in the mesenteric lymph node, there's absolutely no induction of cell proliferation in the absence of SFB, but with SFBs there's very good proliferation shown over here. And in animals, recipients that lack the serum amyloid A protein genes, you can see there still is proliferation, maybe a little bit retarded, but there still is quite good proliferation. There's also induction of ROR gamma T in the mesenteric lymph node T cells. But if we now look in the ileum, in the lamina propria, what we see is something a bit different. And if we now look at IL-17A induction, you see that in these T-cell receptor transgenic cells, many of the cells make IL-17A, but very few, comparatively few of the cells make IL-17A in the absence of serum amyloid A. And we have then learned that this is due to a role of the microbiota, in this case particularly SFB, in activating the type 3 innate lymphoid cells to make interleukin-22. And earlier, Randy Longman in our laboratory had found that mononuclear phagocytes that are monocyte-derived cells that make interleukin-1, interleukin-23, and the TNF family member TNF-SF15 or TL1A, through production of these cytokines, activate the type 3 innate lymphoid cells to produce IL-22, and then IL-22 acts to protect the barrier. Miriam Merad's lab at the same time found that CSF2 or GM-CSF, also made by ILC3 in response to microbiota, contributes to homeostasis and expansion of regulatory T cells in the lamina propria. And what Teruyuki found was that there was induction indeed of IL-22 upon colonization with SFB. And if the animals lacked interleukin-23 receptor, in this case, the IL-23 receptor primarily on innate lymphoid cells, you can see that upon colonization with SFB, there was much less induction of SAA 1 and 2 in the epithelium of these mice. And this is now looking on a log scale at the RNA level here. So this and other kinds of experiments led us to the model that I show here, that upon SFB colonization, there's basically a local short circuit in which the monocyte-derived cells making interleukin-23 act on the receptor in the type 3 innate lymphoid cells. These cells make IL-22 that acts on its receptor in the intestine, in the epithelium, activating phosphorylation of STAT3, and then that leads to expression of SAA 1 and 2 within the first few days of colonization. At the same time, monocyte-derived dendritic cells traffic to the draining lymph node, and they're just polarize the T cells through mechanisms we don't understand yet to become ROR gamma T positive. And these cells now distribute widely throughout the intestine, also outside of the intestine. But it's only here locally where we have serum amyloid A proteins that there is this induction of IL-17, of the effector cytokine. So that's led us to propose then a two-step model in the differentiation and activation of Th17 cells. First, a specification that occurs in a draining lymph node and can be monitored by ROR gamma T expression, and then the induction of the effector function that in this case is dependent on serum amyloid A proteins to a large extent. I shouldn't say completely dependent on that. We think SAA is an example of an endogenous adjuvant that might have a unique or special role on Th17 cells. But this also led us to propose that perhaps endogenous adjuvants like SAA may change the threshold for activation of weakly self-reactive T cells, such as T cells that might be induced by SFB, and that may then lead to predispose animals to autoimmune disease. And that is the hypothesis that we're now following. So we have focused, because of these results, on serum amyloid A, which have been known for many years in the clinic because they are acute phase reactants, much like C-reactive protein. These are apolipoproteins that are associated with high-density lipoprotein and also retinol in the plasma. They're produced mostly in the liver during acute responses to infection. There can be enormous increases in their circulating levels. They're also elevated in tumors, also in synovium of rheumatoid arthritis patients, and in the serum in autoimmune diseases. And it's been reported in multiple sclerosis patients, for example. It's a highly conserved family of genes, and there have been multiple reported receptors, although it's unclear which of them might be physiologically important. Toll-like receptor 2, the formal peptide receptor 2, and also CD36 have all been reported as receptors, and so far we haven't found the function for any of these in what I'm going to show you now. So Junyoung Lee decided to start looking at the role of SAA, which we found works directly on T lymphocytes. And what you can see here is that with recombinant SAA1 and very similarly with recombinant SAA3, he is able to now see a really substantial increase in the proportion of cells that express interleukin-17A, even just in the presence of interleukin-6. And interleukin-6 and TGF-beta are cytokines that can induce Th17 cells from naive CD4-positive T cells. And John O'Shea's group here has looked at other conditions for looking at TGF-beta independent differentiation of cells with IL-6, IL-1 beta, and IL-23. And you can see that in all of these differentiation conditions, adding in recombinant SAA1 leads to this really major increase in the proportion of IL-17 producing cells. And what's really interesting is that if we now look at ROR gamma T expression here, we see that even without any difference in ROR gamma T expression in these cases, we see a big increase in IL-17A, consistent with the idea that this is a second hit, a second signal that activates the effector function of the cell. So Jun decided then to look in models of autoimmunity to ask whether SAAs might be important. And he looked in SAA1 and 2 double knockout mice that we had gotten from Fred de Beer in Kentucky, but he also introduced by CRISPR a mutation in the third SAA gene, SAA3, which is expressed by macrophages in the lamina propria upon colonization with SFB. So he compared in the MOG immunization model of EAE, the wild type and SAA triple knockout mice, and what he sees is really a substantial attenuation of disease, both in the acute phase, as shown over here, but also in the chronic phase of disease over here. And I won't show you the data here, but we know that this attenuation early on seems to be mostly a function of the absence of SAA1 and 2, whereas this more rapid recovery over here seems to be mostly a function of SAA3. And we're beginning to try to understand what might be going on there. So you can see that after immunization with MOG and adjuvant, there's a very rapid increase in circulating SAA1 and 2. This is in the knockouts here for SA1 and 2. This is due to expression from the liver, because you can see both SA1 and SA2 are highly upregulated upon immunization, primarily in the liver, but there's nothing, no activation of SA3 in that organ. On the other hand, looking at the central nervous system, you see no effect of SA1 and 2, but there is induction of SA3, and this turns out to be made by microglia and by monocyte-derived cells that invade the central nervous system after immunization. So what June also noted was that when he looked in the chronic phase of disease, he saw that animals had various levels of disease scores, but also varied quite a bit in their circulating SAA1 and 2. And he was able then to compare the level of SA1 and 2 in the circulation to the disease score, and he saw a remarkably a strong correlation here, that the higher the circulating level of SAAs, the greater the disease score, again, consistent with the SAAs having a role in the disease process. And then he looked at another model of EAE that was developed in Vijay Kuchru's lab using T-cell receptor transgenic mice in which the T-cell receptor is specific for a peptide of the myelin protein, of MOG. So these 2D2 cells can be differentiated in vitro, polarized to become inflammatory Th17 cells with this combination of IL-6 and TGF-beta, and then subsequently interleukin-23. And when they are introduced just into wild-type mice, that leads now to paralytic disease, to the EAE. And he wanted to compare now to SA1 and 2 and 3 knockout mice. I'll only show you results with SA3. There's a mild defect upon introduction into SA1-2 double knockouts. But you can see that introduction into SA3 knockouts almost completely protects the animals from the disease, as shown, both by the percent of animals with disease and the average disease score. And what's also interesting is that gating on ROR gamma T positive cells in animals that were either sufficient or deficient for SAA3, you can see that even though there are ROR gamma T positive cells, even in the knockout mice that go into the CNS, many fewer of these make interleukin-17A. Again, consistent with the idea that there might be a second signal coming from SAA to facilitate production of the effector cytokine in this microenvironment. So now, to put this two-step model in a context of pathogenesis, then, what we think then is that SA1 and 2 in SFB-specific T cells contributes to these cells locally becoming positive for interleukin-17A and interleukin-17F. But on the other hand, in the pathogenesis model of EAE, in the CNS, we see a role for SA3 in the expression of the pathogenic cytokines and the ability of these cells to induce disease. We think that I showed you the example with SFB of these cells going to the lamina propria, where they can be subjected to this kind of induction by SAA. We think that similarly, these cells can make their way to the central nervous system, but it may be by a circuitous route. We don't really know whether they are derived from these cells that are first induced in the intestine, in this case, potentially in the small intestine, but I'll show you in the EAE model, we think it's that they are induced by SAA produced by the liver. And another important question here is, do the Th17 cells that go to the central nervous system need to be cross-reactive with Th17 inducing bacterial antigens? Is there some kind of molecular mimicry involved in the autoimmune disease? And I'd be happy to discuss this after my lecture, but our inclination is to think that that is not the case. So, but we do think that the microbiota are critical in this particular model of autoimmunity, in the MOG-induced EAE. And an example of that is shown here. If we treat animals with either ampicillin or vancomycin, we very effectively protect them from the paralytic disease after MOG immunization. You can see that neomycin and metronidazole do not have this kind of an effect. So it's giving us the opportunity now to begin to look to see which bacteria might be involved here. And I should point out, this is in mice that lack any SFB. This is not an SFB-dependent effect. So just to put this into context then, we think that the microbiota do in fact program the cells to become ROR gamma T positive in this early polarization. Then after immunization with MOG in the presence of adjuvant, there's production of systemic SAA1 and 2. These serum amyloid A proteins act on these primed Th17 cells, activating them, maturing them. They express now the chemokine receptor CCR6 that allows them to traffic to the central nervous system. And once they get there, we think that they can cause some local inflammation, and the positive feedback process activate production of SAA3 by the myeloid cells there. And now the SAA3 acts to generate to induce the pathogenic cytokines, and that leads then to chronic inflammatory disease. So these are all testable hypotheses now to try to understand how these cells make their way from being just primed cells to being actually pathogenic T cells. So what I was telling you then is that we think there is this two-step model of the primed cells and then the effector cells that can be generated by a variety of different means, and these could be homeostatic or pathogenic and contribute to autoimmune diseases. We think that potentially they could be harnessed for either pro or anti-tumor effects. But I'll show you one other example where we think that this two-step model actually is important, and that is a model for autism called the maternal immune activation model. So this particular model was developed after there were reports based on population studies that women who had had infections late in their first trimester of pregnancy were more likely to have children with autism spectrum disorder. And the model that was developed was to either introduce a virus or later just poly-IC, which activates toll-like receptor 3, into pregnant mothers at day 12 and a half of gestation. And that leads then to behavioral abnormalities in the offspring that persist into adult life, as well as to some morphological abnormalities in developing of the central nervous system. This is a model that was championed by the late Paul Patterson at Caltech. And about a decade ago, Paul published a paper showing that this MIA phenotype in the offspring requires that the mother be sufficient for interleukin-6. This paper here in Journal of Neuroscience. This caught our eye because at that time, a really fabulous postdoc in my lab, Jun Hu, had joined the lab, and he had been a graduate student at Caltech. He knew Paul Patterson's work. And we were beginning to work on Th17 cells. This was now really more than 10 years ago. And so we knew that IL-6 is involved in induction of ROR gamma T in Th17 cells. So we proposed that perhaps this might be involved in this animal model, perhaps through induction of Th17 cells. So we got a pilot grant from the Simons Foundation for Autism Research and began to look at this. Now, there are three cardinal symptoms of autism. There are social interaction defects, communication defects, and stereotypic or repetitive behavior. All of these can be modeled in animal models. In mice, it's debatable as to whether they are really related to what's going on in human, but it is what we have available to work with. And what June initially did was to reproduce the Paul Patterson's result showing induction of IL-6 shortly after poly-IC injection, but he also showed then that there is induction of interleukin-17A, and in fact the interleukin-17A in the circulation stays elevated for much longer than IL-6 does. We can block with antibody against IL-17. And importantly, we had made a conditional knockout mouse for ROR gamma T in which we could knock it out specifically in T cells using the CD4-Cre to cross to. And you can see that after poly-IC in these mice, there is no elevation of interleukin-17A in the mother. And he was then able to use this as a tool as well as antibody against IL-17 to see what might occur. Well, one of the first things he showed by doing in situ hybridization was that there is expression of interleukin-17 receptor A in the outer layers of the cortex in the fetus at embryonic day 14.5. You can see it here. And you can see that after poly-IC, there's actually an increase in the expression here. And it is known that IL-17 receptor is regulated in a positive feedback manner by its own signal, by IL-17 binding. And then we're able to start doing these behavioral assays in wild-type mice and mutant mice, in the offspring of wild-type and mutant mice. And this is the marble-bearing assay, which is a repetitive behavior assay in which the mouse is asked to bury marbles in its bedding. And there's a tremendous increase in this kind of repetitive behavior after poly-IC treatment of the mother. And we completely eliminate this if the mother is lacking ROR gamma T in her T cells, specifically in her T cells. Similarly, one can look at a social interaction assay in which mice interact either with another animal here under this cup here or with an inanimate object here. And typically, a normal mouse will spend twice as much time with another mouse than with the inanimate object. And what we see here is that after poly-IC, this is basically a 50-50 sort of ratio, and we basically restore preference for the social interacting mouse if the mother is lacking ROR gamma T in her T cells. And there's also a morphological defect that we observed that is really quite severe, that you see here in the cortex. This is staining with various markers for cells in the developing cortex in the fetus at day 18.5, 6 days after the injection of poly-IC. And again, blocking IL-17 in the mother basically abrogates this abnormality. And finally, June, with his wife Gloria Choi, who is a neuroscientist, together did injections across the uterus into the ventricles of the fetuses, and injecting IL-17A or PBS or interleukin-6. And only after IL-17A injection, there were behavioral defects as well as morphological defects. And in this case, we looked at the behavioral defect as a communication defect, although we also saw these other behavioral defects that I told you about. And you can see the morphological defect as well here. And the last thing I wanted to tell you about is that this requires microbiota, specific microbiota in the mother. And that's where I want to link together the parts of my talk here. Because what June was more recently able to do, and June has his own laboratory now at UMass in Worcester, and he's been looking at mice with or without SFB, or mice that are colonized with a mix of bacteria from Kenya Honda, human bacteria from human that induce Th17 cells in mice, and was able to colonize the mothers and then look at IL-17 induction and the behavioral defects. And you can see here that after introduction of the human bacteria in this case and poly-IC, there's a tremendous increase in IL-17 production without the increase in the introduction of human bacteria, just the black 6 flora from Jackson Laboratory. There's very little IL-17A induced by poly-IC. And this now corresponds to the behavioral phenotypes that I told you about, in this case, the vocalization index, the marble bearing, this is an anxiety assay here, and the interaction assay, social interaction assay. So what we can conclude then is that in this model, the microbiota are absolutely critical in the mother for the priming. And following that priming of ROR gamma T positive cells, a second signal induced by poly-IC allows production of IL-17, which crosses the placental barrier through mechanisms we don't understand and acts on the IL-17 receptor in the central nervous system. I didn't tell you, but the fetus must have intact IL-17 receptor in order for the phenotype to be manifested. And that leads both to the developmental phenotype, the cortical phenotype, as well as the behavioral abnormalities. So this has interesting implications for humans. Obviously, we don't really know whether there's anything like this occurring in humans. There's no question that autism is caused in most of the cases by genetic influence, but there is a known environmental influence, and some of that environmental influence could be due to microbiota. So I pose a question here: do maternal infections cause activation of Th17 cells in a subset of susceptible women, those women who have a particular kind of microbiome that renders their Th17 cells more active, larger numbers of Rho gamma T positive cells? This is not something we can answer very easily, but I hope that this sort of study is going to set the stage for doing more investigation in this area. And I will stop there. This is Jun Hu. The latest work was done by Sang-Du Kim, postdoc in his lab, and all the work on the neurophysiology I didn't show you, as well as histology in the CNS, was done by Gloria Choi, who's now at the McGovern Institute at MIT. We did much of our early behavioral work at NYU with Chuck Hofer, who's now at the University of Colorado. We collaborated extensively with Kenya Honda and Koji Ararashi in terms of the microbiota, and Sang-Won Kim made the conditional knockout mice for ROR gamma T. And these are some of the folks that I mentioned as I went along. Benny Yang is now actually back in China. He was at South Carolina for a while, and he identified the specificity of the SFB-induced Th17 cells. Wendy Wang, now at UCSD, did much of the early work on SAA induction and its role on T lymphocytes. And we collaborated with Rahul on single-cell RNA-seq, Fred De Beer on the SAE1 and 2 mice, and Kenny and Koji on many of the microbiota studies. And I will leave you, I'll stop there and be glad to take questions. Thank you very much.
Well, the hour's a bit late, but we should take a couple of questions, starting right here.
Great talk, Dan. Do you think SAA has direct effects on regulating FOXP3 independent of effects of RR gamma? And sort of second part, IL-10, independent of the ability to modulate RR gamma levels?
SAA, we haven't seen any effect of SAA on upregulation of FOXP3. Is that?
No. I was imagining downregulation.
Downregulation of FOXP3.
You're saying you get pathogenic Th17 cells, but
yeah,
you also...
we really don't know if there's a difference in how SAA influences the homeostatic versus pathogenic. We don't know whether there's a difference between SAA1 and 2 and SAA3 in terms of signaling in the target cells. You could imagine that there might be a difference there. We're looking at that now. All those experiments, I showed you what we know, basically. We can see these effects in vitro that are really dramatic effects. But we haven't looked to see whether SAA in any way inhibits regulatory T-cell induction. And we should probably look at that. Yeah. Right here.
Dan, that was fantastic. I wanted to touch on the question of antigenic mimicry, because as you know, we have some data that actually points to the fact that this may be important. You mentioned that you think that, if I understood correctly, SAA might be changing the threshold of T-cell activation through the T-cell receptor, perhaps. But then later on in your talk, you sort of discounted the possibility of antigenic mimicry. I wonder if you could expand on that. How does that go together?
Well, we have preliminary data that MOG-specific T cells that I showed you in that model, in the SAA3 knockout model, are still dependent on microbiota, even though they are specific just for that peptide of MOG. So it's very difficult for me to imagine that there's some fortuitous cross-reactivity of a Th17 cell inducing bacterial antigen with MOG.
But who says that the adjuvant effect, which is built into gut microbiota, and antigenic effect, if such is present, have to be on the same bug?
I don't think they need to be on the same. So I think they can be separate, yes. Yeah, and the adjuvant effect can be totally irrelevant according to that model, be it in the draining lymph node, joint lymph node, or the CNS draining lymph node, what have you. Yeah. But it's still, it's still a hypothesis as to whether there's a difference in threshold.
Last question over here.
Bringing it down to the clinical realm, there's the well-known syndromes, torch, toxoplasmosis, CMV, herpes, rubella, and now Zika are entering the realm of cerebral abnormalities. And in terms of your hypothesis, in terms of the Th17 in pregnant females, in terms of these syndromes, have there been any specific studies done? Because these would be the perfect targets.
Yeah, I'm not aware of that. And in fact, after we got this result, I called up Tony Fauci a bit over a year ago when Zika was coming to the fore. I thought, my God, this could be it. But obviously, that's not the case. Zika, unfortunately, cannot be. If this was the way Zika worked, it'd be much more manageable clinically, I think. And yeah, it would be nice, but that's not how it works.
Okay.
So, but I'm not aware of anybody really having done those kinds of studies with the other infections either.
Thank you.
Very quickly.
So considering the large pool and the variations of these regulatory T cells in the brain versus the intestine, what is it so acute in the autism? Because that has a tremendous effect, unlike other susceptible tissues. You mentioned about maternal microbiota plus local interleukin-17, but is it good enough to make such a significant impact on total inactivation of the cerebral circuits?
So you're asking, can there be enough IL-17 in the circulation?
No, I mean, is there something else? Because what you see in autism, of course, there is a huge spectrum, but the one with the nonverbal and other issues with a larger brain volume, is there anything else besides the interleukin-17 causing inflammation and causing other effects?
Yeah, all I can say is this is obviously a very, it's a limited model. It's a mouse model. We cannot really extend this to human. There's no question that IL-17 can be sufficient here. Whether it's sufficient in vivo, we don't really know because, when we bombard the ventricle with IL-17, it may not be what's actually happening when the fetus is in utero normally and getting transplacental IL-17. So there could be additional factors. I would agree with that. So we don't really know that. We know it's required in the model. It may be sufficient. And it is just a restricted kind of a model, which even if it is like some human autism spectrum disorder, it may very likely define only a small segment of that spectrum.
Thank you. Outstanding work.
Thank you.
If you'd like to continue the conversation and also enjoy some coffee and refreshments, please let's adjourn to the medical library. But let's thank Professor Littman again.